Chapter 2, Nucleic acid-based cellular activity. So in the cell, there are many cellular activity, and some of them are nucleic acid-based cellular activity. So for example, for the replication, that's the vertical transfer of the DNA, so the genome can be replicated, so that 2 copies, so that's a replication. So this is an example of the nucleic acid-based cellular activity. And then also we have horizontal transfer of genetic materials. So for example, the transcription, translation, and also the recombination between the cell, different cells. So these are all nucleic acid-based cellular activities. And then for the replication, so the first one that we will discuss in this chapter, that's a replication. And then in chapter 3, chapter 4, we will then discuss the transcription and translation. For the replication, so there are 3 different replication hypotheses in the early days. So the first one is called conservative replication. So for the conservative replication, so that means after the replication, so we have the 2 copy of parental strand still stay together, and then we have another double helix of the newly synthesized DNA. That's the conservative. And then for the semi-conservative, that would be the after replication, and then both double-stranded DNA will have 1 copy of the parental strand and 1 copy of daughter strand. It's a hybrid, so that's the semi-conservative replication. And then another one is called dispersive. So basically, the dispersive mean those are newly synthesized DNA and the parental DNA just randomly distribute in both double-stranded DNA. And to confirm the DNA replication mechanism, so in 1958, the Matheson and Stoll, they have designed the experiment to determine the replication mechanism. So what they did is they used the equilibrium density gradient centrifugation experiment. So they demonstrate that the DNA replication is semi-conservative. So what they did is they grew the E. coli in the nitrogen-15 isotope medium, and then transfer many generations so that all the DNA carry the nitrogen-15, then transfer to the nitrogen-14 medium to let it grow for 1 generation, and then they collect the sample, and then for 2 generations, then they collect the sample, so that after the 1 generation of the replication, so they collect the DNA, and then they found the DNA basically after the density centrifugation, they found all the DNA line up in one location, just like this, which should be the 1 hybrid, which should be the nitrogen-15 nitrogen-14 hybrid. And because they observe this phenomenon, so they rule out the conservative replication, because the conservative replication will show 2 fragments. One will be nitrogen-15, one will be nitrogen-14. And since you show the hybrid nitrogen-15-14 1 band, so that just rule out the conservative replication possibility. Then they examine the second 2 generation of replication, and after the 2 generation, and then they found there are 2 bands. One still nitrogen-14-15 hybrid, and the other one is nitrogen-14-14 double helix. So that just confirmed the replication is a semi-conservative replication. And then for the replication, so in the E. coli, basically the chromosome, the size of chromosome is about 4.6 times 10 to the 6 base pair, and the rate of replication is about 1,000 base pair per second. And there's only 1 origin replication, which means there's only 1 location, 1 spot that the in the E. coli, they will initiate the replication. Therefore, the total time required to replicate and divide the cell is about 40 minutes. And then for the human, so the human genome has about 3.4 times 10 to the 9 base pair. And the replication speed is much slower, 20 times slower than the E. coli. And this is because in the eukaryotic cell, the DNA is wrapped around the nucleosome. And so that when the replication occurs, it has to deal with those nucleosomes on the DNA. That's why the speed is much slower. And because the speed is slow and the size is big, therefore there are thousands of the original replications so that they can perform the replication all at once. And then time for cell division is about 24 hours. So let's look at the experiment.
The Messelson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So here, just to demonstrate how they confirm the DNA replication is semi-conservative. Okay. And then for the DNA replication, basically it's a DNA synthesis reaction. So for the DNA synthesis reaction, basically it's the SN2 reaction, because here they say the primer, and then we have template, and then for the DNA synthesis, and for the DNA synthesis, it's very important. It requires a 3' hydroxy group, because this one will attack this alpha phosphate and then release the pyrophosphate and form this the phosphodiester bond between the 3' oxygen and this 5' phosphate, right? So they will form this phosphodiester bond and release the pyrophosphate. So this is the mechanism of the DNA synthesis. And here also another one to show you that incoming nucleotide, and then the 3' hydroxide, the 3' hydroxide will attack this alpha phosphate and then release pyrophosphate and then form this phosphodiester bond. And then the DNA replication model. The DNA replication model, that because we have double-stranded DNA, and then the DNA replication, when we open up this double helix, and both strands will be required to serve as a template for the DNA synthesis. And therefore, there are 3 different types of model. One would be continuous model. So continuous model that explain that the DNA synthesis can go from the 5' to 3' and also 3' to 5'. And semi-discontinuous model, basically, it's both strand will go 5 to 3, 5 to 3. And then another one is discontinuous. It's just 5 to 3, 5 to 3, but every time they can only proceed 5 to 3 and then stop 5 to 3. So they are many 5 to 3. But for this one, the 5 to 3 will be continuously. And then this 5 to 3 will be discontinuously. So this is semi-discontinuous. Okay. And then later on, the scientists identify that the replication model is a semi-discontinuous model. So basically, we have one strand, it's called leading strand. So the leading strand can synthesize the DNA 5' to 3' continuously. And then the other strand is called lagging strand. For the lagging strand, there are many Okazaki fragment. Each fragment is about 1 to 2 kb long. And so this part also synthesize 5 to 3, however, discontinuously. And then this mechanism can allow both strands to replicate in the 5' to 3' direction. Okay, the direction of this fork movement, the replication fork movement, is 5 to 3. And here is just another diagram to show you the semi-discontinuous DNA replication. So when you open that double helix DNA, and then one replication fork will move to the right, the other replication fork move to the left, right? And then we have 5' to 3'. That's a leading strand continuously. The green one here is leading strand, and the red one is the lagging strand. And they both replicate the 5' to 3', and then also the general direction will along this the replication fork. So let's look at the video, another video.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork.
So in the next slide, we will discuss all the enzymes that involve the DNA replication. So helicase, basically, to initiate the DNA replication, then you need to open up this double helix. So to open up this double helix, we need the DNA helicase to open up this double helix.
The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process.
The reason we need the primase is because they're going to form this RNA primer, as I mentioned in the previous slide. For the DNA synthesis, it requires a 3' hydroxy group to attack this the alpha phosphate of the incoming nucleotide. So that's why we need a primase to synthesize RNA primer to provide this 3' hydroxy group to initiate this SN2 reaction.
This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA.
So in here, you see the two different types of DNA polymerase involved in this process. The first one basically is DNA polymerase 3. So it's the main polymerase to synthesize the DNA. And then here, after the exonuclease remove the RNA primer, then the DNA polymerase 1 will come in to fill up the gap.
Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So that's the DNA synthesis. Okay, and then in this process, so here is the list of the enzyme and protein that are required for DNA replication. So first of all, we need a DNA helicase. The helicase basically will bind to this lagging strand because it will run from 5' to 3' to open up double helix. And then when you open up double helix, it will bring in the positive supercoil. Positive supercoil ahead of this replication fork, as we discussed in the chapter 1. Therefore, it requires this DNA gyrus to remove the positive supercoil. And then when you open up the double helix, we need the SSB, which will be the single-stranded binding protein, to protect, stabilize the single-stranded DNA. And then the primase. So for the primase, to synthesize a few nucleotide RNA so that we can initiate the replication. And once we use the DNA polymerase to synthesize new DNA, then the exonuclease will remove these RNA, and so that the DNA polymerase 1 will come to fill the gap. Finally, the DNA ligase will repair the phosphodiester bond between these DNA polymerase 3 synthesized DNA fragment and DNA polymerase 1 synthesized fragment. Okay, so here is the list of the protein and the enzyme that participate in this DNA replication process. So in the next few slides, basically, we'll explain each of the enzymes function as we just discussed. All right, so here we have helicase and single-strand binding protein, and so here they require the ATP so that the helicase will be able to open up a double helix, and they bind the lagging strand because it can run from 5 to 3 direction, and this is a template for the lagging strand. Okay, and the gyrase. So for example here, when you open up double helix, you introduce the positive supercoil ahead of the replication fork. Therefore, you need a topoisomerase 2. As we discussed in chapter 1, the polymerase II will cut both DNA fragment and then move around so that to remove the positive supercoil. Okay. And primates provide a 3' hydroxy group to initiate the DNA synthesis. All right, and then we have the exonuclease or RNSH that can remove the RNA primer. And then polymerase 1, 2, 3. So as I mentioned, the main polymerase is DNA polymerase 3, responsible for the DNA synthesis. And the DNA polymerase 1 basically now be the one after you remove RNA primer, then the DNA polymerase 1 will come in to fill in the gap. Okay, and then the role and the function of DNA polymerase 2 still uncertain, but it might involve in the SOS DNA repair. So here is just to explain further, the DNA polymerase 3 can synthesize a new DNA strand. And DNA polymerase 1 is the one to fill in the gap that when we remove the RNA primer. And then for DNA polymerase 1, there are two subunits. One is called clinal fragment. The other one is 35 kilodalton fragment. And then for the clinal polymerase has a 3' to 5' exonuclease activity. And then 35 kilodalton has 5' to 3' exonuclease activity. So 5' to 3' exonuclease activities be like to remove the old strand and synthesize a new strand. If it's a clinal, then you will not be able to remove the strand 5 to 3. The 3 to 5 basically is the activity to check the incorporated nucleic acid is the correct one or not. Basically, it's a proofreading activity to check the correct nucleotide. And then, so as we mentioned, the DNA replication, we have leading strand and lagging strand. Although, if you look at the detail, they are in a different. They basically synthesize DNA in a different direction. However, in general, overall, the DNA replication has been coupled. Therefore, they will move in the same direction from 5 to 3 as the 5 to 3 of the leading strand DNA. Okay, so the movement is coordinated to allow the polymerase on both strands to travel together in the direction of the replication fork movement. Okay. So basically here just the general it's the schematic diagram to show you that the coupling of the DNA replication on the leading strand and lagging strand in bacteria basically is achieved by physically association. The physically associating the protein replicating each strand into one large protein. We call it a replication complex or replisome. So the replisome basically associates with the DNA, and this replication complex move together as the replication fork move. Protein within the complex can engage and disengage from the DNA cell, but remain associated with the fork. So in the E. coli, the multi-subunit assembly of DNA polymerase sliding clamp and the clamp loader is called a DNA polymerase 3 holoenzyme, or we call it a replisome. So it contains 2 copies of the multi-subunit DNA polymerase 3, which the polymerase 3 has the alpha subunit. The alpha subunit, which is a DNA polymerase activity, has the DNA polymerase activity. Epsilon subunit basically have the 3' to 5' proofreading exonuclease activity, and the theta subunit, which is stimulated exonuclease activity. And together, these DNA polymerase 3, together with the sliding clamp and the clamp loader, and the clamp loader basically continually reload the sliding clamp on the lagging strand, and so that they can coordinate these activity together so that they can move move these ribosomes together as the replication fork move. Okay, here is just more detail about each subunit that you can see that, for example, here is the lagging strand, this is leading strand, we have polymerase 3, the coenzyme here, and basically DNA polymerase 3, then we have here's the helicase, DNAB helicase, and then we have clamp loader, and then we have the sliding clamp. They move the general direction to this direction, but each of the detail is different direction, opposite direction. And just like here is more detail. So the top one is the leading strand. So the polymerase 3 core will move this direction, and the helicase, DNAB helicase, will also move this direction. And then we have this clamp loader, and then this is the lagging strand. Okay, so they will also replicate this direction. Okay, replicate this direction, and here's the single strand binding protein. So that's the general picture of the replisome. And here we can watch another video to show you the replication fork coupling.
During DNA replication, the DNA molecule is duplicated to generate two identical copies. The correct copying of DNA is of vital importance to the cell, as errors that are introduced into the DNA may lead to heritable mutations that could be deleterious to the cell or organism. Thus, elaborate machinery is used both to assure correct copying and to allow the process to be regulated. Coupling of the two DNA polymerases, one for each strand, allows the process of fork movement to be coordinated. To understand how polymerases are coupled at the replication fork, we will first show a standard representation of the replication fork. In this typical representation, two DNA polymerases are moving in opposite directions, with one copying the leading strand of the DNA and the other copying the lagging strand. To expose the single-stranded DNA at the replication fork, a six-membered ring-shaped complex called DNA helicase first separates the parental DNA strands. This separation gives the DNA polymerases the access they need to allow copying. The new DNA strand is always made in the 5' to 3' direction. Because the replication of the two DNA strands is coupled, the leading strand is copied continuously, while the lagging strand is copied in short stretches called Okazaki fragments. Each Okazaki fragment is initiated by a primase that synthesizes the short RNA primer. This primer is later elongated by DNA polymerase. The single-stranded DNA between the newly made Okazaki fragments is bound by single-stranded binding protein, or SSB. In order for the two polymerases to move together with the helicase as the fork moves, the DNA template for the lagging strand must be looped around so that both polymerases are moving in the same direction. With the polymerases now coupled, DNA replication can proceed coordinately on both the leading and the lagging strand. Once the lagging strand polymerase reaches the RNA primer from the upstream Okazaki fragment, the lagging strand loop is released, and Okazaki fragment maturation and ligation by polymerase 1 and DNA ligase occurs. While this maturation is occurring, the primase enzyme that is also travelling with the fork as part of the replisome initiates a new RNA primer on the lagging strand. Once this primer is made, the lagging strand polymerase on the replisome, having completed synthesis of the previous lagging strand, then re-engages with the 3' end of the new primer, forming a new loop on the lagging strand. DNA synthesis thus proceeds with the leading strand polymerase continuously engaged with the DNA, and the lagging strand polymerase engaging and disengaging as each Okazaki fragment is synthesized. To synthesize the lagging strand, the DNA template is looped out while it is coupled to the replosome complex, and the loop is released after each Okazaki fragment is completed. The length of the looped DNA at its maximal size is therefore the length of an Okazaki fragment, which in bacteria is about 1,000 base pairs.
So that's the process of a coupling replication. So we just discussed the coupling replication fork, and now the replication patterns. So as we know that most eukaryotic and the bacteria DNA, they replicate bidirectionally, and then the DNA under the control of the original replication is called a replicant. And then on the right side, you can see on the right side, that's a typical circular E. coli chromosome. Circular E. coli chromosome has a single replicant because it replicates from a single starting point, the original replication. And the DNA replication begins with the creation of a bubble, which is a small region where the parental strand have separated and progeny strand have been synthesized. And then the two replication fork arise from this bubble and move in opposite direction. As the bubble expanded, the replicating DNA begin to take on the theta shape until both replication fork meet on the other side of the circle. And then on the left side, that's eukaryotic chromosome. As we know, the eukaryotic chromosome is very long, and they have many replicants. And then the replication in this replicant begins simultaneously. And then they move bidirectionally until they meet each other. And also until they reach the end of the chromosome, which is the telomere. So previous slide, we discussed this regular bidirectional replication fork. And now it's another replication mechanism. It's called rolling circle replication. So sometimes the bacteriophage, they will adopt this rolling circle replication pattern. For the rolling circle replication, basically you would need to have the 3' hydroxide. You need to have a nick on the positive strand of DNA, and so that you provide this 3' hydroxide group as the starting point to initiate the DNA replication. As they extend from the 3' end for the replication, as you can see, the green arrow here, and then the parental strand is peeled off, and then they complete the replication as a circle, and then this blue one has been peeled off completely, and then the RNA primer will be generated to initiate replication for this one. So that's the rolling circle replication. Let's look at this video about the rolling circle mechanism of plasmid replication.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. a region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.
All right, so this is the typical example of a rolling circle replication. And now let's watch the next one. Now let's watch another video, which is the plus RNA replication. And one of the examples of the plus RNA virus would be the SARS-CoV-2 virus. And here is just to show you a typical bacteriophage, how they infect the E. coli, and then replicate the plus RNA replication.
Some RNA phage, such as MS2 and Q beta, contain only a single positive plus sense strand of RNA. They infect only F plus strains of E. coli because they attach to the pilus. A positive plus strand of RNA is one that can act directly as messenger RNA. A negative minus strand is complementary to the plus strand and cannot act as messenger RNA. To replicate more positive strands, the positive sense RNA strand must first be used to synthesize an unusual phage-induced RNA polymerase. RNA synthesis from an RNA template does not normally occur inside the uninfected cell, and so the entering plus RNA must code for the synthesis of this RNA polymerase, which is also called RNA replicase. The RNA replicase uses the positive sense RNA as the template for synthesis of negative sense RNA, and the negative sense RNA is used to synthesize more positive strands. The positive strands serve as messenger RNA for synthesis of phage coat proteins and are then incorporated into the phage during assembly. A phage-induced lysozyme lyses the cell, releasing the phage to the environment.
The termination of a DNA replication occurs when the two replication fork moving away from the single origin of replication meet on the opposite side of the circle in the circular chromosome of bacteria. When the replication forks approach each other, the replication complex disassemble and the two growing strands join, and this results in the interlinking of the two new daughter DNAs. The two circular DNA molecules are separated by topoisomerase. And sequences that are involved with termination are called tersites. A tersite contains a short 23 base pair sequence, and the termination sequence are unidirectional and can be recognized by a unidirectional contra helicase. This helicase known as the TAS, which is the replication terminator protein in E. coli. This protein can recognize the consensus sequence and prevent the replication fork from the proceeding. And in E. coli, there are 10 tersites, and binding of a TAS protein to this tersite prevent replication fork from passing through by inhibiting helicase activity. So that's the termination of the prokaryote replication. This is just an example of a bidirectional replication of the circular E. coli genome. Produce catenated molecule that are separated by the topoisomerase 2. In E. coli, it's a gyrase, right? Interlinking circle, you see they can be separated by the topoisomerase 2, which is the gyrase in E. coli. This figure just to show you there are 10 tersites and 5 on each side, and this replication fork, when they move down, you can see the blue in the front. That's permissive the complex, so that the replication fork can access these tersites until they reach this third side, which is the origin in the front, this is non-permissive, then it will stop. And also on right side, also the replication fork can access all these third side until they reach this orange non-permissive conformation, and then will stop. That's how they terminate the replication. So the mechanism of eukaryotic DNA replication is very similar to that of prokaryotic DNA replication. However, eukaryotic DNA replication requires special consideration due to the differences in the DNA size, because eukaryotic DNA, the genome is much bigger than the prokaryotic DNA genome. And also it's a unique linear DNA, and the structure we call it a telomere. It's not like the E. coli, it's a circular DNA, there's no linear DNA end structure. And also a distinctive DNA packaging complex in the eukaryotic, because in eukaryotic DNA, in eukaryotic cell, we have the nucleosome, which pack the DNA with the histone, and when the replication occur, it has to go through this nucleosome structure. So that's why it's more complicated than the prokaryotic cell. So the DNA replication in eukaryotic cell is a highly regulated process, and usually require extracellular signal to coordinate the specialized cell division in different tissues of multicellular organism. So the eukaryotic chromosome and in distinctive sequence we call as a telomere, and the telomere basically is a repeated sequence with about 10 to 20 kilobase pair long, and the repeated sequence is like a TTGGGG, and this complementary sequence of this repeat sequence will be AACCCC, and the end of the linear DNA fragment, when we have the replication reach the end, that will have a problem for the leading strand. For the leading strand, there's no problem because they can just the DNA polymerase can replicate the sequence all the way to the end. But for the leading strand, since there's a RNA primer, when you remove the primer, then that create a gap, and that will shorten the leading strand, and the chromosome length will become shorter and shorter after the replication. So in the human, the telomere are repeat end sequence, and this repeated sequence is about 15 to 20 kilobase pair long. And so when the replication reach the end of the telomere, basically there will be a telomerase try to extend the leading strand, so that the lagging strand will use those extended sequence to create an RNA primer, so to replicate the telomeres area, so that the DNA will not be shortened after the replication. That's a replication of telomere. And is the mechanism of the replication at the telomere region, and with the help of telomerase. As I mentioned, the telomerase basically will try to extend the leading strand. Once you extend the leading strand, then the RNA primer can be created in the lagging strand, so that we can replicate the lagging strand sequence. And now we will watch the next slide, which is a video of the replication in the telomere region.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction, and adds additional repeats as it moves down the parental strand.
So this is the leading strand.
The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
So this is the mechanism of the replication at the telomerase region. And then based on this, the mechanism of DNA replication and the several drugs has been designed that can prevent the DNA replication and to control some disease. So inhibitors of the DNA synthesis, so that can be used in the laboratory in the treatment of bacteria, virus, and the neoplastic disease. So these kind of the inhibitors can be divided into 3 main classes. So for example, they can be used to prevent or reduce the synthesis of precursor, like the nucleotide, or they can be used to affect either the template or the priming ability of the growing strand, and also those act directly on polymerase or other enzyme needed for the replication. So for example, inhibitors acting on DNA polymerase and other enzyme of DNA synthesis include the aciclovir, aviditicoline, aminocoumarin, and camptothecin. So these are the examples that the scientists developed that can be used to prevent the disease through the control of the DNA replication. And then the precise copying of the genetic information during the DNA replication provide the genetic stability to the living organism. However, the error can be introduced during the DNA replication. So normally the DNA polymerase, they should carry the proofreading mechanism, and this proofreading mechanism basically is from the 3' to 5' exonuclease activity, so that they can proofread whether the nucleotide is correct or not, and so that we can reduce the error to the minimum, like once in every 10 to the power of 9 or 10 to the 10 base pair after the replication. And then of course some the error still occur, and when the error occur, that can introduce the DNA mutation. And so the next few slides we will discuss the DNA mutation and how the cell repair the DNA mutation. So basically the DNA mutation can be the point mutation or the insertion and deletion. So the point mutation, that means the one nucleotide has been changed, and that is a point mutation. and it can be a transition or a transversion. Transition basically is in which one purine or pyrimidine is replaced by another. Transversion would be a purine is substitute for a pyrimidine or vice versa. So these two are possible kind of a point mutation. And then the point mutation basically can have three different types of the end result. So I can cause the missense mutation. So the missense mutation basically change one nucleotide that change the amino acid. So in this example here, UAU to the UCU, so that change from tyrosine to serine. That's called missense mutation. And the second type of the point mutation would be nonsense mutation. Nonsense mutation basically you change one nucleotide and then that will change the amino acid to the stop codon. So in this case, UAU to UAA. That's a nonsense mutation. And the third type of the point mutation is silent mutation. So the change one nucleotide and then the amino acid is still the same, the coded the same amino acid. That's the silent mutation. So these are three different types of point mutation. And then for the insertion deletion, that will be the frame shift mutation, because you either insert one nucleotide, two nucleotide, delete one nucleotide, two nucleotide, and that can change the codon because they change the reading frame. In this case, frame shift, if you have base addition in between these two A, then you shift this reading frame one nucleotide, so that change all the amino acid. If you delete one nucleotide, and that will shift the reading frame. So these are the frame shift mutation. And also there's also another type of mutation if you add or delete the three nucleotide, although that will not change the mutation. But if you have many add this triple addition, that will cause some mutation. So here is some diseases caused by the trinucleotide repeat. So for example, Fragile X syndrome is CGG repeat, and then if you have the more than 230 repeat of CGG, that cause the Fragile X syndrome, that's a mental retardation. And then Huntington disease is a CAG triple nucleotide repeat. If you have more than 36 triple repeat, that will cause the Huntington disease. So these are the example of the trinucleotide repeat mutation. And then the tautomer shift. So basically, although the nucleotide, the nucleic nitrogenous base are chemically quite stable, the certain hydrogen atom bound to the base are able to undergo the tautomerization. That means they change their location on the bases. So for example, in the adenine here, the hydrogen atom here attached to the nitrogen, but now they can shift to this location. So that change, that's called a tautomer shift. So that change this the whole structure from amino structure to immuno structure. But the majority of the adenine cytosine, they adopt this amino structure. For the guanine and thymine, the majority of guanine thymine, they adopt the keto structure. And then you can see the hydrogen ion here from the nitrogen and shift to the oxygen here, right? Become the hydroxide from here to here, from here to here. So that's tautomer shift. But the tautomer shift can cause the structural change, and that will cause the mutation. So in this case here, so if we have this tautomer shift of the A, and now after the replication, the A will base pair with the C. That's the tautomer. A tautomer, they no longer base pair with T. instead base pair with the C, and then after the second round of the replication, that will introduce this CG mutation. So this is tautomery shift to cause the mutation. And the large-scale mutation, that will be the chromosomal mutation, so that can have the translocation, inversion, deletion, and the non-disjunction. If a mismatch was introduced in the DNA, then there's a nucleotide excision repair system that can repair the mismatch repair. So for the mismatch repair, then the mut-S protein will scan the DNA sequence and bind to the mismatched bases, the mismatched base. Then the mut-H protein, which is the endonuclease. The mut-H endonuclease recognize the methylated DNA sequence of GATC, so that means this is parental strand. And then the mut-L link mut-H and mu-S. The link mu-H protein will nick the opposite strand of the methylated A base on the parental strand. Then the helicase UVRD will come in to unwind the DNA from the nick and proceed past the mismatched nucleotide. Exonuclease cut away the nicked strand, and single strand binding protein stabilize the resulting single strand. The excised DNA region is resynthesized by DNA polymerase III, and the DNA ligase seal the nick to form the double-stranded DNA. So this is the mismatch repair to remove the mismatched nucleotide. And DNA methylation. So basically the bases that are most frequently methylated are the guanine and cytosine. So basically for the methylation, so in here you can see the cytosine. Cytosine, if you are methylated 5' methylcytosine, then it can, after the deamination, then be look like a thymine. So that's a conversion from C to T. Therefore, the base pair from CG then to AT. That's a DNA methylation. Now let's watch the video.
This is a video of a 3D animation on the mismatch repair pathway in E. coli. In the cytoplasm is the E. coli's DNA, the molecule that contains all of its genetic information. DNA is made up of nucleotides, each of which contains a phosphate sugar backbone and an organic base. The two strands of DNA are held together by hydrogen bonding between complementary base pairs. Adenine pairs with thymine and cytosine pairs with guanine. DNA mismatches occur during DNA replication. For simplicity, this animation will show DNA as a flat molecule. Before replication can begin, a helicase needs to unwind the DNA molecule. In E. coli, DNA polymerase III catalyzes the incorporation of new complementary bases to the new DNA strand. This is only the alpha and beta units of the DNA polymerase III enzyme. However, it is possible for DNA polymerase III to incorporate a non-complementary base to the new strand. This is known as a mismatch. A mismatch could result in a faulty protein being coded for, so it needs to be repaired. The first step in the mismatch repair pathway in E. coli is recognition of the mismatch by the mut-S homodimer. Mut-S recognizes the mismatch and binds to it. Then, the mut-L homodimer binds to mut-S, ready for a later step in the mismatch repair pathway. Meanwhile, up to a kilobase away, the mut-H protein recognizes a GATC site. Mutate is a weak endonuclease, which will create a backbone incision between the guanine and adenine residues on the newly synthesized strand when activated. Mutate is able to differentiate between the newly synthesized strand and the template strand by the lack of methylation markers on the newly synthesized strand. This is why the pathway is also known as methyl-directed mismatch repair. In order for mutate to become activated, it needs to bind with mutel. This causes the DNA to bend. The newly formed backbone incision allows an exonuclease to enter and begin excising nucleotides from the newly synthesized strand. The exonuclease excises the mismatched nucleotide and stops excising just beyond it. Now, DNA polymerase III can begin to resynthesize the new strand, inserting the correct complementary base where the mismatch used to be. The final step is for DNA ligase to ligate the backbone nick. The mismatch pathway is now completed, and all the newly synthesized DNA is complementary to the template strand.
So this is the mismatch repair and the mismatch. Now the next section we will discuss is a DNA damage. So DNA damage can be caused by a physical agent or the chemical reagent. So the mutagen and mutagenesis, so basically the physical agent and chemical reagent that cause mutation are called mutagens, and include the environmental chemical and ionizing radiation. And this process that produces a mutation is called a mutagenesis. And mutagenesis can be induced by a variety of chemicals, but also happens spontaneously as a result of the faulty DNA replication and the base modification, such as the tautomerization and deamination of base. So for the base modification, that could result from the base analog or alkylating agents. So base analog are the mutagenic chemical that can substitute for the normal purine or pyrimidine during the nucleic acid biosynthesis. So for example, the nucleoside analog bromodeoxyuridine, the BRADU is formed when the 5' here, the 5' the bromouracil, 5' bromouracil is chemically linked to deoxyribose. The 5-bromouracil, 5-BU, is a derivative of the uracil, and its 5' position of the pyrimidine ring is halogenated. Like here, you can see here. If a 5-BU is incorporated into DNA in place of thymine, the tautomery shift will occur and the 5-BU will base pair with guanine. Like here, instead of adenine, then we'll base pair with the guanine. If shift from keto form to the eno form. Okay, this is a 5-bromouracil. The keto form and eno form. And alkylating agents. So alkylation is the process in which the electrophile attack the negatively charged DNA molecule and add a carbon-containing group called alkyl group. Okay, so in this case here we have this S-methane EMS, which is the alkylating agent, right? And then you have the GC pair, guanine, cytosine, and then we'll bring in the methyl group here, and so then this is called O6-isoguanine, and then it's no longer base pair with cytosine. Instead, it's base pair with thymine, so that will change from GC to AT. That's alkylating agents. And the damage caused by the radiation, it could be the UV radiation, ionizing radiation, byproduct damage. So the UV radiation that can cross-link the adjacent pyrimidine on the same DNA strand, forming the two major lesions. So the vast majority of the lesions are pyrimidine dimer. So basically the two carbon on each of the two pyrimidine form a cyclobutyl ring. That's the UV radiation. Okay, see this UV radiation, ionizing radiation, that including the gamma ray and X-ray interact with the DNA molecule in a similar manner to the UV ray. They cause the damage by ionizing the molecules surrounding the DNA, especially water. Other form the free radical, which are extremely reactive, especially those containing the oxygen. As a result, they can attack the neighboring DNA molecule and change the base or cause a single double-strand break. So for the damage repair, then we have single-strand damage repair. The single-strand damage repair, that including the excision repair system, and also you can have a direct reversal of damage or damage tolerance. Okay. For the reversal, the undoing damage. So for example, here the photoreactivation or light repair, because there's an enzyme called photoreactivating enzyme, or the photolyase can be activated by absorbing the energy from the near UV to a blue light to break the bond holding the two pyrimidine. That the thymine dimer caused by the UV exposure can be undoing through this process, light repair. Okay. And another one is the suicide enzyme. So basically, alkylation of the O6 of guanine can be reversed by the O6 methylguanine methyltransferase. So you can see this. There's an enzyme, and then here's the O6 of guanine alkylation. Then they can remove the methyl group. Once they remove the methyl group, then the enzyme become inactive. So that's why it's called a suicide enzyme. That's a DNA repair through undoing the damage. For the single nucleotide base excision repair, we have this excision repair system. In this system, we have the DNA glycosylase that can recognize the damaged base. And then the DNA glycosylase can cleave the glycosidic bond between the nitrogenous base and the deoxyribose, and release the damaged nucleotide and forming the base site. The base site means you only have the sugar phosphate but no nitrogenous base. Then the AP endonuclease will recognize the base site, and the AP endonuclease will remove this the phosphodiester bond in the backbone, and then a number of addition residue on the 5' side of the AP site. And finally, the DNA polymerase will come in to fill the gap and ligase and repair the phosphodiester bond. And that's the video to show the base excision repair.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine dimer or other defect to replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, when adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone.
So
The
this is the
result
first
is an
step
intact phosphodiester backbone
to cleave the
missing a
nitrogen
base.
space.
The site is called an AP site.
And then to
In
form
this
the
case,
AP
it is
side.
missing a purine, and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone at AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick, and the repair process is complete.
So this is the example of the base excision repair. And then the nucleotide excision repair that involve the basically this is common for the DNA lesion caused by the UV radiation, oxidative chemicals, reactive oxygen species, cross-linking agent, and the intercollecting anti-neoplastic drug. Now, because these can distort the DNA structure, and the incision enzyme system recognize the strand with the bulky damage and make a cut on either side, removing an oligonucleotide containing the damage. In the E. coli, and this system involved the UVR ABCG proteins. These are the key incision enzyme system responsible for cutting the damaged DNA and producing an oligonucleotide that is 12 to 13 base pair long. So normally the UVRA dimer recognize DNA damage and bind to the damaged site with the UVRB in an ATP-dependent manner. The DNA strand at the site of the damage are separated by the helicase activity of the UVRB, and the UVRB stably associate with the DNA, while the UVRA dissociate. And UVRC, which has the endonuclease activity, is then recruited to the UVRB DNA complex and cut either side of the lesion through the nick on both 5' and 3' side of the lesion. And the action of the UVRD is to release the damaged DNA strand, and UVRC and the UVRB find the DNA polymerase, fill the excised region, and the DNA ligase seal the nick completing the repair system. The nucleotide excision repair in the eukaryote is similar to the prokaryotic system. And here's the video to show you the nucleotide excision repair.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
So here, this is just the nucleotide excision repair. So until now, we discussed the reversal and the excision repair, and next would be the damage tolerance. So both direct reversal and excision repair system are true repair process because they remove defective DNA entirely. And however, sometimes cells they do not eliminate the defective DNA entirely, but simply skirt around it. This phenomenon is referred to as the damage bypass mechanism or damage tolerance system. And here we have this damage bypass mechanism. It's called a recombination repair. So basically, you can see if we have a pyrimidine dimer and the replication machinery skip over these pyrimidine dimer and resulting in a gap on the daughter strand, and then through the recombination, this daughter strand, through this recombination, then the original gap can be filled by this recombination. So this is the damage repair, damage tolerance system. So another one is called SOS response, and this is like an error-prone repair, because when a bacteria is subject to the extreme condition, and then a great deal of DNA damage occur, then it will have the SOS response, and so that will be the error-prone repair. The SOS response is a multifaceted cellular response in which more than 40 genes are induced when DNA is damaged. In this response, the REC-A coprotease activity is activated by the UV damage or another mutagenic treatment. followed by the cleavage of a transcriptional repressor, LEX-A repressor, and this will cause inactivation of LEX-A protein. As a result, DNA polymerase is formed and become active. Then the DNA polymerase employ the error-prone bypass of the region by synthesizing the nucleotide across from the DNA region, even though the damaged region cannot be read correctly. The polymerase can efficiently bypass three most common types of DNA region, for example, like pyrimidine dimer, UV light, and the AP site. Without proofreading, mutations are introduced in the repaired DNA strand. So this error-prone repair is the main cause of UV-induced mutagenesis. So this is the SOS response. When the both strands of DNA are broken, the result is called a double-strand break. And double-strand break is the most dangerous form of DNA damage. And the DNA double-strand break can be repaired by two main mechanisms. Number one, homologous recombination, HR, and number two, non-homologous end joining, NHEJ. The HR, homologous recombination repair system, is more accurate than the NHEJ system, since it uses the homologous DNA sequence and the system chromatid to repair the damaged region. And here's just the example to show you the homologous recombination in the E. coli. Basically, the REC BCD homologous recombination pathway begin with the introduction of a double-strand break, and then the REC BCD protein bind to a DNA double helix stranded break and use its DNA helicase activity to unwind the DNA toward a so-called chi site, which has been found on average every 5 kilobase in the E. coli genome. The REC BCD protein can produce a single-stranded tail, and which is then coated by the REC A protein, and this will allow the tail to invade a double-stranded DNA duplex and search for a region of homology. This creates a displacement loop. It's called a D loop. Once the tail finds a homologous region, a nick occurs in the D loop DNA, possibly with the help of REC BCD, and this nick allows the REC A and the SSB, single-stranded binding protein, to create a new tail that can pair with the gap in the other DNA. And the DNA ligase seal both nick to generate a Holiday junction, and we call this as a Harvard chiasmus and the chi structure. And the branch in the Holiday junction can migrate in either direction simply by breaking old base pairs and forming a new one in branch migration process. And this can help to recombine the DNA and repair it. So that's the homologous recombination. And then for the non-homologous end joining repair system, the process occurs simply by the rejoining of the broken DNA end and does not require another template duplex such as the sister chromatid. And this repair is an error-prone repair because of a lack of a template. It can occur at any time in the cell cycle. Therefore, the NHEJ is the only readily available repair pathway for cells in G1 phase of the cell cycle. Because most mammalian cells spend the majority of the time in G1, therefore the NHEJ is the predominant mode of double-strand break repair in the mammalian cell. And the NHEJ process in bacteria and eukaryotes require a heterodimeric protein Q7080, dimer, and now can bind to the broken end of the DNA to protect the DNA ends from the degradation and to recruit several other proteins to promote the synapsis of the region with homology. Subsequently, the Ku's helicase activity is activated through the phosphorylation, which then can promote the unwinding of the DNA end, followed by the base pairing of the homology region in the DNA end, leaving the flaps composed of the end of the other non-pairing strand. The phosphorylation is activated by the binding of a protein kinase, DNA protein kinase, to the Q7080, and finally the flaps are removed by the nucleus, gaps are filled in, and the DNA strands are ligated together. So that's basically the process for the NHEJ.